Classical genetics began with an elegant but rigid blueprint. When Gregor Mendel crossbred the garden pea plant, Pisum sativum, he observed traits that fell into distinct binary categories, round seeds or wrinkled seeds. Mendel defined a biserial allelic framework, assuming a single gene locus is occupied by one of exactly two alternative configurations, a dominant allele or a recessive allele. 20th century cytogenetics redefined the gene as a long, continuous strand of nucleotides. Physical mapping of chromosomes showed that a single locus spans thousands of base pairs, providing the molecular space for more than two variations to exist. Because a gene is a linear sequence, spontaneous mutations, such as point mutations or frame shifts, can occur at distinct positions or sub-loci along the nucleotide strand. This molecular plasticity creates a diverse spectrum of allelic variants within a single population. Despite this variety, these alleles exhibit locus homology. They represent alternative structural configurations of the exact same physical coordinate on a homologous chromosome. This proximity explains the absence of classical recombination. Because these variants occupy the same site, crossing over does not occur between members of the same allelic series during meiosis. Viewing the gene as a vast sequence of nucleotides rather than an indivisible particle provides the necessary foundation for understanding multiple allelism. Analyzing this complexity requires a distinction between the individual and the population. A standard diploid organism can harbor a maximum of only two alleles at a specific homologous locus, one inherited from each parent. In contrast, the collective population gene pool harbors an indefinite repertoire of alleles, denoted by the variable n. This principle is a staple of advanced genetics examinations. Consider a diploid insect species where a single gene locus controls 12 distinct wing pattern variations across a population survey. To account for these 12 variations, the wider population pool must contain a minimum of 12 distinct alleles interacting with one another. However, because the species is diploid, any single insect remains strictly limited to possessing exactly two alleles at that specific locus. This demonstrates how wild phenotypic polymorphism is generated collectively at the population level, even while individuals obey the structural limits of meiotic segregation. The first major evidence of this population-level diversity came from the human ABO blood group system. Carl Landsteiner discovered blood clumping in 1900, but it was Felix Bernstein who provided the mathematical proof in 1924 that a single gene locus, the I gene, possessed three distinct alleles, IA, IB, and lowercase I. This system introduced codominance, where IA and IB alleles are expressed simultaneously. Both alleles independently produce their respective antigens in a heterozygote. They interact with the recessive I allele through complete dominance. A single dominant A or B allele completely masks the I variant. These biochemical interactions yield precise phenotypes used to determine parentage probabilities and forensic markers. The ABO system proved that multiple alleles interact through complex hierarchies, moving beyond the simple dominant-recessive pairings of early Mendelian models. Thomas Hunt Morgan provided the second pillar of evidence through Drosophila melanogaster. In 1910, he isolated a mutant fruit fly displaying white eyes instead of the standard wild-type red. Morgan tracked this trait to the X chromosome, identifying a sex-linked inheritance pattern. Males are hemizygous for this locus, expressing whatever single allele occupies their 1X chromosome. The white gene codes for an ABC transporter, moving tryptophan and tyrosine precursors, essential for synthesizing the fly's eye pigments. A null mutation at this locus blocks the entire transport pathway. Even if downstream brown and scarlet genes are functional, no pigment is made, making the white mutation epistatic. Morgan's team discovered many mutations at this site are hypomorphs, partial loss-of-function variants reducing transporter efficiency. Panning to the summary notes, this creates a graded spectrum of eye colors. These alleles follow a linear dominance hierarchy based on pigment deposition. This demonstrates that multiple alleles can produce a quantitative scale of a single trait based on the efficiency of the resulting protein. In plant biology, Edward East and Albert Blakesley identified multiple alleles as a mechanism for enforcing genetic diversity within tobacco plants. They discovered the S gene locus, which contains dozens of unique self-incompatibility alleles identified as S1, S2, and S3. These alleles regulate pollen-pistil interactions. If an S allele in a pollen grain matches the pistil tissue, a biochemical arrest prevents the pollen tube from elongating. Fertilization only proceeds when the pollen carries a mismatched S allele, ensuring the plant cannot pollinate itself. This system uses multiple alleles to mandate cross-fertilization, preventing inbreeding and driving genetic variation across the population. The S allele series shows that multiple alleles can function as an active biological strategy to promote and preserve diversity. Across humans, insects, and plants, these systems share the property of functional coherence. Every allele in the series modifies the same specific physiological pathway. This property is used to solve non-complementation problems, such as those found in the June 2021 CSIR net examination. When two different recessive mutations are brought together in a single organism, they are in a trans-heterozygote configuration. If the resulting phenotype remains mutant, it indicates functional non-complementation. Both copies of the gene lack the functional nucleotide sequence at the same locus, leaving the organism unable to produce the wild-type protein. This failure to restore the normal phenotype proves the two mutations are alleles of the exact same functional gene. Non-complementation serves as the molecular diagnostic for locus homology. It distinguishes multiple alleles of one gene from independent mutations occurring at different epistatic sites. Given the pressure of natural selection, we must ask why one highly efficient allele does not eventually displace all other variants in a population. Polymorphism is actively preserved through balancing selection, a set of mechanisms that prevent genetic drift from erasing allelic variety. One mechanism is heterozygote advantage. In the case of sickle cell trait, individuals carrying one normal and one mutant hemoglobin allele possess a higher survival fitness than either homozygote due to malaria resistance. A second mechanism is frequency-dependent selection. Here, an allele's fitness increases as it becomes rarer in the population. In plant S allele systems, a rare allele succeeds more often because it is less likely to encounter a matching incompatible tistil. Many other mutations are neutral, having no impact on survival. These variants fluctuate randomly through genetic drift, persisting in large populations without selective pressure. High levels of genetic polymorphism are not random occurrences. They are mathematically stabilized biological configurations. Population genetics allows us to quantify these evolutionary forces and predict how they change over time. To calculate the number of distinct genotypes possible for a gene with multiple alleles, we use the formula total genotypes equals n multiplied by n plus 1, all divided by 2. This formula is essential for solving population genetics problems, such as deducing allele frequencies from a sample of individuals with different ABO blood groups. Determining the frequencies of the 3 ABO alleles requires an expansion of the standard Hardy-Weinberg equilibrium. Biologists apply the trinomial expansion formula, p squared plus q squared plus r squared, plus the 3 corresponding heterozygote frequencies, totaling exactly 1. By mapping these variables to observed phenotypic counts, such as matching the codominant NEB group to the 2pq term, the frequency of each individual allele can be isolated. These mathematical models transform raw field data into precise measurements of allelic distribution. By tracking these frequencies across generations, researchers can monitor how mutational variants and selection pressures fluctuate within a living population.